Hello, here we're going to focus on the morphology of cell responses to damaging stimuli. So specifically, we're going to look at cell injury as well as cell death. And for this, we're going to focus on apoptosis and necrosis, comparing and contrasting the two. So let's first look at the morphology of cell injury. So here we have a simple drawing of a normal cell, and typically what happens when there is pathologic stimuli, the cell undergoes some morphologic changes indicative of injury. Many of these changes will be appreciable on electron microscopy. Not all will be able to be discernible on light microscopy. So the most obvious change is that the cell actually increases in size, so it swells, and often this is due to accumulation of water. There may also be some changes in the cell membrane, such as blebbing, because a lot of the time the membrane is damaged in cell injury. In addition, the mitochondria can also exhibit swelling, and this is not really appreciable on light microscopy. Perhaps the most obvious change that we can actually appreciate on the microscope is hydropic change, which is the accumulation of water in the cell, leading to swelling of the cell. Another organelle that may be affected is the endoplasmic reticulum, and this may undergo dilatation as well as detachment of ribosomes. This is really not visible on light microscopy. In addition to these features of cell injury, sometimes metabolic derangements can also give rise to intracellular accumulation of various materials. And some of these may actually be visible on light microscopy. Now these can be sort of divided into endogenous versus exogenous materials. A good example of an endogenous material is lipids, for example, fatty change in the liver, and this is due to accumulation of triglycerides in the hepatocytes. This may be related to alcoholic liver disease, toxins, and sometimes even hypoxia. This can be due to increased entry of lipids into the liver, or defective metabolism, and even export of lipids, so the decreased lipids leaving the hepatocytes. Another important endogenous substance that can accumulate would be abnormal proteins. Another good example will also be in the liver, when you can have Mallory bodies accumulating, and these are cytoskeletal proteins. Other accumulations include hemosiderin, iron material usually found in macrophages, associated with the breakdown of blood. So this is often seen in association with hemorrhage, and lipofuscin, which is sort of the wear and tear material in senescent cells. Now, in terms of exogenous material, there can be ingested substances such as carbon, for example, in lung macrophages or alveolar macrophages in smokers, and also certain microbial products can also accumulate within the cytoplasm where the cells are actually unable to get rid of them. Now moving on to cell death, two main types, necrosis and apoptosis. So let's first look at necrosis. Cell size is usually enlarged or increased, so it's sort of like a progression from cell injury. The nuclei can undergo various changes. They can become pycnotic, which is very shrunken with very dark chromatin. They can undergo karyolysis, which is sort of just fading away of the chromatin, or karyorrhexis, where the nucleus actually breaks up into little pieces. So you can see like fragmentation of the nucleus. And eventually the nucleus will disappear, and that's how we recognize necrotic cells under the light microscope. Now the plasma membrane is also abnormal in necrotic cells. There can be blebs as well as disruption of the cell membrane, and as mentioned earlier on, the cell contents undergo denaturation of the proteins. Sometimes the cells will appear a little bit more densely eosinophilic because of this, and there may be enzymatic digestion of cell contents. Now, necrotic cells tend to incite an inflammatory response. So what happens is often the phagocytes will come in, and also neutrophils, and they will try to mop up all the necrotic material. Apoptosis is a little bit different. Cell size often will actually be decreased. So instead of the cells swelling, the cells will shrink. The nuclei can undergo similar changes that we see in necrosis. There may be pycnosis, shrinkage with condensation of chromatin. There may also be karyorrhexis, which is like a fragmentation of the nucleus. And you can see little black dots sometimes, or very dark blue dots of fragmented nuclear material within the cell. Generally, the plasma membrane and cell contents tend to be more intact. However, they tend to break up into smaller pieces, and these are known as apoptotic bodies. So these are still bound by plasma membrane. In terms of inflammation, generally apoptosis does not really incite an inflammatory response. However, there will be macrophages that will come and eat up the apoptotic bodies. The mechanisms are a little bit different. So for necrosis, it's very similar to cell injury, which we talked about earlier. For example, with membrane breakdown, loss of membrane integrity, ATP depletion, etc. Now for apoptosis is a bit more of a regulated process, and there are two main steps. The first step will be initiation, and this can be either intrinsic, which starts off at the mitochondrial level, or it can be extrinsic, which is mediated by death receptors on the cell membrane itself. Following the initiation step, then we have the execution phase, and this primarily involves a series of caspases, and these enzymes are actually ultimately responsible for breaking down the nuclear material as well as the cytoskeleton. So one more thing which is important to note about apoptosis is that it can be physiologic, maybe as part of normal growth and development, and also hormonal cycles. In contrast, necrosis is always pathologic, and we've talked earlier on about the pathologic stimuli that can give rise to necrosis, things like hypoxia, physical agents, chemical agents, biological agents, etc. Now, going back to apoptosis, some examples of physiologic conditions would include immune cell deletion of certain clones of lymphocytes that would otherwise have reacted with our autoantigens. So this process tends to break down in autoimmune disease. This is why our immune system actually attacks the body's own antigens. Another example would be in the endometrium, for example, due to hormonal influence, progesterone, estrogen, etc., you have cycling of the endometrium. Apoptosis can also be seen in pathologic conditions. For example, in certain infections, like in hepatitis B or hepatitis C infections, the liver cell death occurs through apoptosis. And another example would be in patients who are receiving chemotherapy. This can give rise to very severe DNA damage and can cause cell death through apoptosis. So just to summarize, we've looked at the morphological features for cell injury. Most of these are appreciated only under electron microscopy, but we can see some changes like swelling due to hydropic change, and we can also appreciate some intracellular accumulations. For cell death, the two main types, necrosis versus apoptosis, we've gone through some of the differences. Essentially, for necrosis, the cells would generally swell, the cell membrane will be damaged, and it does incite an inflammatory response. Whereas for apoptosis, the cells tend to shrink, they break up into smaller pieces, and they don't generally incite an inflammatory response. We've also briefly looked at mechanisms for apoptosis and also the pathologic and physiologic causes. You will also see that there are many types or patterns of necrosis, and these are very well described in your lecture notes, as well as in some of the talking pods and talking slides in other parts of this web chapter.